Yep. Very good. Well, thank you to the organizers, certainly for the invitation. And I hope I can contribute to the third keynote address of some really very interesting papers so far. And I have to say that what we just heard in Parkinson's will be a very interesting mirror and reflection of what was noted in the presentation, and that is the key term neuroinflammation. And we'll talk about how this all comes together through this paper that we'll be presenting here from Keystone Bio on our further development of a clinically effective biotherapeutic against Porphyromonas gingivalis, which is quite a mouthful. It's a gram-negative bacteria with incredibly unusual properties, and I'll speak more to that. I'll just call it PG for short. And really what we're doing at Keystone here is looking to modify inflammation-driven diseases from cardiovascular to dementia to one of its most common types, Alzheimer's. And this does include Parkinson's, which we'll talk about. So Porphyromonas gingivalis, we wrote recently in JAD. We consider a new term that we coined, biological extremophile, and its role in systemic inflammatory disease, including Alzheimer's. I hope to review the first clinical trial results and the in vitro characteristics of the monoclonal antibody that's being developed specifically for KB001, which is a murine IgG1. And then the subsequent development of an analog, an IgG1 human chimeric of the mouse monoclonal to address future issues with repeat use in xenoproteins. And then a little bit discussion on the further clinical development pathways for the biological. PG has really, in its evolution, solved a major amount of issues, both in its location and its environmental location and the environmental stressors associated with that, which is the oral cavity and the host defenses which are present in the oral cavity. Other than H. pylori, which is another unusual, we call considered an equivalent biologic extremophile that was identified in the stomach and ultimately became the etiologic agent for gastric hyperacidities and ulcers, and ultimately then a series of transformative cancers of gastroadenocarcinomas and gastric carcinomas. This organism lives in the mouth, in the subgingival sulcus primarily, which is pretty anaerobic. It has to solve wide pH fluxes, wide oxygen tensions. It's solved wide temperature fluxes. It defends itself from over 1,200 microbial proteins on the innate side. Interestingly, it's a very small population, that's less than 5% of the overall total oral microbial population. And at some point, there's a conversion from a more normal PG to a virulent form of PG that then modifies the polymicrobial biofilm environment and actually recruits these organisms to assist in its survival and contribution to other disease producing bacteria that co-associate with itself. So what seems to be a bacteria that eventually gets a foothold in your gingival sulcus and is sitting there, ultimately the additional bacteria that it recruits under its management is what ultimately leads to the additional severe periodontal disease and gum disease, but it itself would not contribute immediately. So there's an interesting complex relationship between it and the additional bacteria that are recruited. It's very unusual in that it has no known bacteriophages. Of course, these are viruses which transfer all kinds of virulence factors in and out of bacteria and modify certain behaviors. None have been identified, which is highly unusual and about the only case that I think anyone has ever seen in the bacterial community. The important aspect here is that when you get infected with this chronic lifelong infection, we know of no individual which can clear the infection or has cleared the infection naturally. It defends itself from both, of course, the innate and adaptive immune systems. And if one is not impressed by, these are gene profiles and gene networks, each of these being separate gene complexes, it has solved all of these to maintain a chronic, lifelong active infection. So basically, its operation for disease over time is eventually there's an infection, localization in the mouth, forming the polymicrobial biofilm. Depending on age, we'll review this. 40 to 80% of adult mouse may have then PG. It most importantly was part of the discovery of our Keystone Bio team over the last 8 years, which led to the convergence really for the company and the solutions to it, is that these bacteria release an inordinate amount of outer membrane vesicles that are packed with very complex toxins. They have their own proteome and they have their own genome. And these then travel from the localized vasculature in the mouth and lymphatics through the bloodstream, ultimately ending up in various large vessel, small vessel, end vessel organs, and of course, end organ disease, such as we see here, that include neuroinflammation of the systemic vasculature, the brain, the heart, the liver, pancreas, joints, etc. And so what we really have is a 1.5 plus nanomolar amounts of these coursing through the patient who's infected with these literally daily for as many years as they carry and are alive. As you might imagine, the systemic inflammation is very detrimental over time. And it's not an acute process. And actually the biomarkers of inflammation that are activated, given this release of this exotoxin vehicle, one would think that you would have high fevers and be very endotoxic, like shocked. But there's quite a mystery as to why these don't really induce that. They induce certainly a hot inflammatory profile in your blood, but for the most part, it's like silent hypertension. You don't realize that you're suffering from the presence of these over time, which is unusual. We're not used to, of course, dealing with diseases that don't present themselves sometimes with overt symptomatology. We believe that we had a groundbreaking release of a paper which was pulling together data and a new hypothesis, which is really starting to become more mainstream here now in the neuroinflammatory field. And that was an explanation for how these outer membrane vesicles became and are the major driver for neuropathogenesis and neuroinflammation. It ties the cholinergic hypothesis, iron dyshomeostasis, and the salivary lactoferrin issues together in Alzheimer's disease, unlike any other convergent etiology has done before. Now, what we see over time is generally somewhat after adolescence, when the oral microbiome, the normal healthy oral microbiome, begins to be damaged by smoking, drinking, things like that, that of course adolescents, people do over time, bad nutritional choices, et cetera, you can see that it's almost a decade, sort of by decade, increase in the prevalence of the organism. And just to give you a visual of what I was speaking about, here's a biofilm here, a complex biofilm. Here are the organisms, the cells themselves. And if you look closely and blow those up, you'll see this bacteria is studded with these very large and numerous blebs. And these are the outer membrane vesicles that are pinched off in a very regular manner over time, and are actually very important communication vesicles for the bacteria, as well as host defense, nutrient and energy sources. So this is absolutely, the bacteria depends on being able to produce these. Just to give you a little better picture here, it's a really nice transmission electron micrograph of these structures. They're highly organized and are made by a very specific multi-gene, about 12 to 16 genes are involved in their creation structure of what's called the type 9 cargo secretions protein system. And then these, of course, are released, and they have very specific protein and lipoprotein and nucleic acid moieties that are put into them. The proteome of these at current count is 151 proteins. So as you might imagine with systemic vascular inflammation like this, we now know that these are proven contributors of vascular inflammation, both now in cardiometabolic, and very recently, we now believe we have causation for the early cognitive decline, neuroinflammation that leads to early, middle, and late dementia and end-stage sporadic Alzheimer's. It may be that now, if we just look at the impact of this agent, it may be the cause for 30 to say 50% of Alzheimer's disease by the organism at this point. A paper that we're putting together right now, and a very exciting comment to be able to be made now after many years of research, both by our groups and other groups. As you can see here, this is an example of blood inflammatory biomarkers that are all turned up. These are certainly not the kinds of biomarkers you want in your blood for any length of time, not to mention days, weeks, or months. And then we've got a list where there are very solid, large numbers of papers that show the very tight association now with a large list of neuroinflammatory and degenerative disorders, and of course, vascular systemic disease. In all of the infectious disease work I've done over the last 30 years in my life, I've never seen anything quite like where there has been a organism which has set up a particular disease process that has such a wide array. We believe this is one of the, if not the largest, missed unknown inflammatory agents discovered to date. So let's talk about what we're doing to solve this problem and the biological activity of the biotherapeutic. The approach taken some time ago was rather straightforward, but to do minimal disruptions of the whole bacterial cell, and not knowing, of course, what we really needed, but to look for down at the end, effective down screening or end screening for an effective antibody at the end of the day. So disruptions of a whole PG was done. Immunizations, mouse monoclonals were made. Somewhere on the order between 500 and 1,000 monoclonals ultimately had gone through this system. Dental plaque, in this particular case for screening of hundreds of patients were used. And the idea there was to just have an antigenically relevant target from many, many cases. And then that was used to screen any of the antibodies that could bind these hundreds and hundreds of patients. So we were looking for a rather common and conserved antigen that could be displayed. There was literally one out of the many that looked very interesting in terms of its binding properties by immunofluorescence and this kind of a down screening. That was moved into its first clinical trial. It was a simple placebo-controlled blinded phase 1. And in this case, 14 patients with periodontitis who had PG were root planed, cleaned, essentially went through a standard of care for dental cleaning, and then were given at that particular time standard metronidazole treatment to suppress detectable PG. The patients are divided randomly then into two groups. One, of course, that's receiving the antibody subgingivally, and the other one is a saline control, as that was the vehicle that the antibody was delivered in. And then the actual treatment schedule itself was 3 micrograms of the antibody or saline per tooth was administered to all teeth in the subgingival sulcus, as you can sort of see here. Very fine, small layering of the fluid along the subgingival gum line. No injections, just topical subgingival oral application. And then there were a number of things done, but most importantly to show you was what happened to the PG bacteria. As you know, leads to a chronic lifelong infection after the treatment. So these are the direct number of counts at the beginning of the trial. Patients with the antibody are the boxes, triangles are the controls. And as you can see, most remarkably, out to 9 months, and this is in the stacked plot, all of these patients that received the monoclonal antibody had no recolonization of PG and only one breakthrough at 12 months. It's quite an astounding outcome. But because the trial was set up at that particular time for a periodontal outcome, which they didn't understand the complexity of the disease, it was considered having missed its primary endpoints and was sort of missed in general, both in the field and literature. Importantly, additionally, 22 lab strains, another 105 clinical isolates. We then derived a phylogenetic tree for the complete family of PG and selected PG that represented across the phylogenetic tree. All of them have bound our monoclonal to date. Monoclonal antibody resistance studies, I won't show you the data, have also been done. And preliminary results are that there's no antibody resistance escape that we have been able to determine. And most likely that's because it's directed to multi-gene protein targets. Here we see the antibody's effect on the biofilm. And what we are excited about is a relatively low, from 0.1 to 10 microgram concentrations. We see between 90 and 99% inhibition or interruption of reformation or formation of this is in this particular case, a monospecies biofilm. Once we took a closer look at where the antibody target engagement was on the bacterial surface, we found, as you can see here in a gold labeled SEM photomicrograph here, that we get a very interesting dotting of the monoclonal antibody. And when we look closely, of course, these are the emerging outer membrane vesicles that the bacteria has to produce to survive, feed itself, and protect itself. And when we purify those outer membrane vesicles, we, of course, see that in addition, they avidly bind the outer membrane vesicles, essentially binding the released toxic cargo as well as that on the bacteria itself. Iron's an important source. This is another example on how our antibody does interfere with its ability to take in iron in a hemagglutinin type assay. So we were then interested in identifying, of course, the target. The target looks like it is a highly conserved part of the type 9 cargo secretion system that I mentioned. This has to have an orchestrated endoproteolytic catalytic activity that has to come together with 3 enzymes, and those have to work together in concert to create these structures, which ultimately allow the bacteria to release these vesicles. What's very interesting is this hemagglutinin adhesin complex of the type 9 cargo secretion system has a triple repeat in it. And so each one of those is able to bind the antibody, contributing to its biologic activity. So this complex of gingipanes and Hag that are secreted on the surface basically have to allow itself to auto-catalyze itself to produce these structures. And of course, this repeat epitope antibody, as we described, interferes with and locks that process down. The binding of the antibody, the murine monoclonal to this target is nanomolar, so it's really pretty decent, pretty tight. And one of the issues that were of concern was that the antibody could be cleaved in the proteolytic environment of the bacterial infection in the mouth. And most importantly, the mouse monoclonal is not cleaved. So we were, of course, happy to see that, and may have also contributed to its clinical success. The manufacturing has been completed, and the drug substance and product is ready for clinical trial, which should begin with patient recruitment in July this year. The toxicity testing, it was essentially unremarkable, up to 100 times clinical effective doses, including IV treatments and also including wound healing, gum healing testing. So with the idea that this treatment may be needed or repeated at 6 months to 12-month intervals, depending on breakthroughs for particular patients or for people for the rest of their life, there was a need to develop what we felt was a human chimeric due to the xenoprotein nature, suggesting that over time, exposure to the mouse monoclonal may in fact lead to local inflammation. You may see anti-drug antibody to limit biological effectiveness, and you might see, of course, additional systemic reactions to the murine monoclonal. So in preparation for that, we constructed a molecular design to derive a human chimeric IgG1 with, of course, the variable heavy and light change from the mouse. We derived 16 plasmids to ultimately screen. We screen these based on equal potency and competition, solid phase assay, target assays against the target, and then did a pull down from these 16 to, I believe the initial down screen was to 8. Ultimately, we selected a subset of 4. We tested these now again in anticipation of cleavage by the human monoclonal, as it had already been reported in the literature that antibody is cleaved, human antibody is cleaved by the organism in that setting. Not surprisingly, our lead candidates were cleaved relatively efficiently. We anticipated that outcome, and then went ahead and did some very detailed mapping of the cleavage site so that we could identify a site-directed mutagenesis approach to engineer out that substrate cleavage site, to which we did successfully. And as you can see here, all of the various forms of the hybrid of the human monoclonal were made resistant to the PG cleavage. And down selection of these is now down to one specific lead, and that is our H5 from these others, H7, 8, and 14. This is compared to the parent monoclonal here, and somewhat interestingly, we ended up with the down selection of this one due to the fact that we had a 10 plus fold or 100-fold, somewhere between 10 and 100-fold increase in binding affinities. So we're now moving the monoclonals into a second phase 1/2 randomized double-blinded placebo trial, so that we can determine the effectiveness and the oral clearance on vascular inflammation. So again, as I mentioned, the drug substance is completed. The IB and final regulatory approvals are wrapping up, and patient selection, as I mentioned to you. We're going to look at expanded efficacy dosing to see if we can simplify the dosing and tolerability of the topical application. We also expect that we'll be able to get metronidazole out of there, since metronidazole in the control group in the first trial did not show any effect. So most likely we'll get the antibiotic out of there and be down to just the monoclonal antibody. And then, of course, we'll investigate in much more detail the clearance of P. gingivalis, time to reoccurrence again, the effect of the treatment on the vascular markers of bioinflammation at screening of 1, 3, 6, and 12 months. And then a little bit additional endpoints and secondary endpoints will be to look at what's happening in the microbiome and the changes that go on into the microbiome during this time. We'll be also looking at anti-serum gingipane and anti-gingipane toxin antibodies, which are formed, as I mentioned, to the outer membrane vesicles. And these, in addition, form immune complexes in these patients, adding to the disease process. So this sort of what the development schedule looks like for both of these antibodies, both in vascular inflammation and neurovascular dementia cognitive type trials over the next 24 months. The pipeline that's developed around this is also very exciting. We've got the biologicals, as I reviewed here, and what's not reviewed is a companion diagnostic, which is really important to tie these two in for specific diagnostic, treatment, management, and outcomes. And that is a series of 3 companion diagnostics to measure the toxic vesicles, to measure the disease-associated antibodies, and most importantly also is to identify PG that carries the virulence gene signature, because as I mentioned, not all do. And so we don't want to, of course, diagnose, misdiagnose, and then treat someone who doesn't have the virulence gene issues and the toxic issues in them. Ultimately, with this information that we have, we are in a very good position to go very preventative and then ultimately develop a PG vaccine based on this antibody to this very conserved structure. All of this work is, of course, covered by a large patent profile, an array of submitted and provisional patents that you see listed here, both world and USA filings. And with that, I wrap up and show you here the many associated collaborators that we have as CROs, academic, and the like, and particularly for this study, focus on this group, which has been important in the work that I presented, and particularly thank Jan Pontempa and Katie, Garth, the various teams from these organizations, which have allowed us to get this work done. With that, I thank you for your time and I'll take questions.